all my time.
It's my pleasure to welcome Joe Miano, the J. Harrison, MD, Distinguished University Chair in Vascular Biology. Dr. Miano has degrees in biology and exercise physiology and experimental pathology. In addition, Dr. Miano and I spent time together at the MD Anderson Cancer Center, Eric Olson's lab, who is on this call. Dr. Miano is one of the more accomplished scientists I've had the pleasure of interacting with. He's also inspired me to be fearless and work extremely hard. What a great role model you were. I think I'll stop embarrassing you there. And Dr. Miano, if you could start sharing your screen, take it away.
Thank you, Dr. Staudinger. Staudy. So, I'm really happy to, so it's a pleasure to be here and to tell you about our work on CRISPR and more recently, the prime editing. All the work most of the work I'm going to show you, I should say, is unpublished. Some of it's old unpublished, but therefore you haven't seen it. And I'm looking forward to having a nice, hearty discussion about this awesome technology. So I want to start, as I sometimes do, in the beginning. So you all recognize this, the most numerous DNA-containing entities on planet Earth, that is bacteria and viruses that infect bacteria, bacteriophage, and very numerous. And by more than 10 to 1, the phage outnumber bacteria. And so there has to be some homeostasis here. We need bacteria for the ecosystem to work. And nature's evolved these awesome technologies, or I should say defense mechanisms that have been adapted for technologies. And you'll appreciate the CRE-LOX from bacteriophage. Of course, innate immunity gave us restriction enzymes, which is absolutely essential for molecular biology. And then there's this interesting adaptive immunity that's known as CRISPR. And it's a long history, but we've been CRISPRized for quite a while because unbeknownst to us, the yogurt, among other dairy products that we enjoy so much, only come about because of CRISPR. And this was proven by Rodolphe Berenguer in 2007, a beautiful science paper where he basically proved the importance of CRISPRs in bacteria to ward off the invading bacteriophage. And so nature's been very kind to us in that regard. But it was these two towering women of science, Jennifer Doudna on your left and Emmanuelle Charpentier on the right, who recently, as you all know, you should know, won the Nobel Prize in Chemistry. And this was the paper that is the basis for that award. This was published in 2012. I remember it well because I heard Jennifer speak about this about a week after it came out at Cold Spring Harbor. And after I picked up my jaw off the ground, I had a nice conversation with her about the amazing applications of this. But as is a really good paper for the young people to read. It's pretty simple, I have to say. Looking back, it's been 8 years. But the basic finding was they harnessed what was known in bacteria, and they developed it for, in their hands, in vitro DNA cutting on demand. And what was striking to me, and still is the last sentence of their paper, and you can read it in yellow there, where they're proposing to use this new CRISPR technology for genome editing in a very elegant way. And the work, oh gosh, I just realized I don't have the most recent version of my talk, but that's okay. So the work led to this revolution. So in 2012, when that paper came out, you may remember zinc finger nucleases and talins. They were the method of choice for editing cells and animals. But as soon as this paper came out, an avalanche of papers followed, and movies came out, bad movies like Rampage with The Rock, good movies which just came out, Human Nature. I highly recommend that. That's on Netflix. I just saw it for the first time recently. It's beautifully done. But CRISPR has really eclipsed these other genome editing platforms, and everybody has been CRISPRized. So how does this work? So in its simplest form, as the science paper described, it is a two-component system. The beautiful work they did was to combine two RNAs into a single guide RNA, shown in purple and red here, that's programmable. And the other component, of course, is the nuclease. Think of it as a restriction endonuclease without activity, and the activity isn't manifest until the guide RNA brings that endonuclease to the molecular address where the double strand break occurs. And then nature has these repair systems necessarily in all our cells to repair these double breaks, and that's the non-homologous end joining that will create a number of outcomes, and they're random, although recently there's evidence to suggest that it's not as random as we think. But in any event, a number of outcomes occur, and though it's not terribly accurate, it's much more efficient than the three-component system, which is our favorite platform, because here the third component is a repair template, so we bias the repair for this ectopic template that has a substitution according to whatever one wishes. So you see this little red nucleotide change, that would be a single nucleotide substitution, and that'll be a theme a little bit later. And so here, this is very precision-guided editing, and so this can be used for a variety of methods, and for today's purposes, it's for making very subtle substitutions. So it's not as efficient as the two-component platform, but it's much more accurate. So CRISPR Ease, and it's so simple now, the only thing one really needs to contend with is the design of the CRISPR RNA, which is the programmable part of this single guide RNA that the Doudna-Charpentier paper so elegantly provided for us. So this is our favorite program for designing that CRISPR RNA. And then once you have that, it's submitted to a company like Synthego, and then you just wait for your synthetic guide to come into the mail with the Cas9 protein, and then you order your oligo from IDT with your substitutions, and you're off to the races. So simple is this platform that is literally globally changed how we're living now and how we'll live forever. So CRISPR has, particularly in the agricultural world, has really fundamentally changed food, wheat that no longer rots, apples and tomatoes that can stay on the store shelf for weeks. There's the insect-borne diseases like malaria that will be tackled in our lifetime, perhaps. There are 50 clinical trials now at the NIH for somatic genome editing. And then there's the very thorny issue of human germline genome editing, which I'm not going to talk about at all today, although I will highlight the recent publication from the commissions of the National Academies. It's about a 250-page read, but it's worth reading because it's really some recommendations about how we should proceed with altering basically our species. So we do germline genome editing, but we do it in the mouse, and that's really the basis of my talk. So usually when I'm talking about CRISPRizing mice, I'm touching on a number of applications, but I really want to just laser focus on one aspect of how we use CRISPR in the lab, and that relates to the non-coding portion of the genome. And why is that? Well, you may know that DNA, our genome, was often referred to as a genome comprised mostly of junk, right? And this was first proposed by Susumu Ono in the late 60s. He was a very smart man. He knew the size of our genome, but he was way off with referring to most of it as junk, because we know from ENCODE and a number of other sources that much of the non-coding genome is functional. And so I want to do something a little different, and some of you may think this is hokey, but I think it'll really make an important point. This is a page of your genome, one page. There's 4,300 characters here. This is actually the Calponin locus, which I started working on when I was in Eric Olson's lab 3 decades ago. What you see here in red is the first exon encoding Calponin's protein. So that's coding sequence. Everything around it, the other 4,200 odd nucleotides, is non-coding sequence. So the amount of coding sequence on this page is almost exactly the amount of non-coding sequence in our entire genome, which is about 1.5% to 2%. So here's something to think about, and you'll probably remember this more than anything. If we want to print out the entire coding portion of the genome with the same font, both strands on double-sided Xerox paper of normal thickness, how much paper would that entail? Okay, so here's a perspective. So this is the Statue of Liberty. Everybody recognizes that. But on the bottom left, if you look carefully at the very bottom, at the platform of the statue, you'll see people. And if we printed out just the coding portion of our genome, it would amount to about 4 feet of paper. All right, so less than the average height of a teenager. Now, in this sub-instruction manual are the instructions for, well, not the instructions, but the coding sequence for about 20,000 proteins and the mutations that we're very familiar with: Duchenne's, sickle cell, cystic fibrosis. So I refer to these as low-hanging fruit. Most of the protein coding, if not all, have been identified. Most of the coding mutations have been identified, and there are therapies underway. There's clinical trials with CRISPR and other platforms to combat these genetic diseases. So now, what about the non-coding portion of the genome? So if we printed out both strands on two-sided paper at the font I just showed, what would that look like? Well, and I want you to imagine this the next time you're on Liberty Island. It would be more than 2/3 the height of the Statue of Liberty. Right? So in this previously referred to as junk DNA are a number of things. I just list a couple broad categories. One is a growing list of non-coding genes, long, short, circular. And two, millions and millions of snippets of DNA called transcription factor binding sites. And that's what I'm going to be talking about for the rest of this talk. One last point here. I refer to this, all this sequence as high-hanging fruit. Very difficult to identify nuggets of information that are important. But I'm up for a challenge, and I've been struggling with understanding just one transcription factor and its binding site for the last quarter century. And it stands to reason just by the sheer volume that most disease mutations lie in this dark matter of the genome. So here's the transcription factor that I've been working on since I was a graduate student in the 80s, the late 80s, serum response factor, SRF. Incredibly conserved protein. Many transcription factors are. Even invertebrates, fly and worm, have very few substitutions. And this serum response factor binds a sequence known as a CArG box, which is a 10 base pair snippet of DNA shown below. And that's a sequence logo of more than 3 million CArG boxes. I'll get back to that number in just a while. So we and a host of other labs, including EO, have knocked SRF out in a number of organs, and it's absolutely essential for life. You cannot live without SRF. There's no redundancy. There's no backup paralogs. If SRF is mutated or deleted, it generally spells death of the organism. So gastrulation in the complete knockout of SRF in every cell, mice do not gastrulate. And CRE drivers that knock SRF out specifically in these different tissue types all lead to death or severe problems. So very important transcription factor. That's all I have to say about SRF. The rest of the talk is basically about its binding site, the CArG box. Oh, one more thing. So SRF, how does it do this, right? So there's, as you'll see, there's millions of these transcription factor binding sites known as CArG boxes. So SRF dances around the genome and lands on these, and it's really an anchor. It doesn't have a lot of intrinsic transcriptional activity, but what it does have is the ability to interact with, as of today, 76 different cofactors, a few of which are shown here. And so, for example, for SRF to direct the smooth muscle differentiation program, it must work with myocartin to direct that specific program. And in the face of disease or some growth stimulus, it will toggle off of myocartin and its CArG boxes to another cofactor, ELK1, and new CArG boxes to drive a new program of gene expression. So it's a real interesting toggle switch whose specificity is conferred by these cofactors. Now I'm done with SRF. So what we did many years ago was we mapped all the theoretical CArG boxes in the human and mouse genomes. And so this is a screenshot of the human genome. It's about an 8 million base pair window. And what you see at the top is the fruit of our labor. So those in black are non-conserved CArG boxes, theoretical CArG boxes, and those in red are conserved in mouse, at least mouse, and usually many more mammals as well. And in the middle, you'll see some of our own unpublished ChIP-seq data from human coronary artery smooth muscle cells in blue and human umbilical vein endothelial cells in red. And you will see other screenshots like this as I go through the talk. So what we're able to do is we're able to leverage this information, the CArGome and the binding site, the activity, to discover new SRF target genes. And so that's what we've done. So several years ago, we discovered this gene called LMODI-1. Well, it was already discovered, but it really was understudied. And this LMODI gene is highly restricted to smooth muscle lineages, as you can see in this GTEx portal screenshot here. On the left are vascular smooth muscle cells and bladder, in the middle are GI, and at the end is the uterus. So anything that's smooth muscle restricted, as EO knows, I'm very interested in. So we got very interested in LMODI-1. It has a number of highly conserved CArG boxes, and I want to focus on just the one on the two on the right, because conventionally, the way things have been done to interrogate the activity of these binding sites is to hook them up out of context to a reporter, in this case beta-galactosidase, to look at its activity. And so as you can see here, the wild-type promoter of LMOD1 containing these CArG boxes drives expression of LMOD in a manner we'd expect based on the middle panel showing LMOD1 protein in this embryo. But when the two CArG boxes are mutated, and this has been done in many labs, including Eric's, the activity is abolished, right? So that was the traditional approach. But we wanted to know, are they really necessary in their native genomic milieu? And so what we did was we CRISPRized them. So this was an experiment we did several years ago, 3-component CRISPR, where we're using, of course, the guide RNA in gold, and the Cas9 not shown here, and the repair template, a 135 nucleotide single-strand oligo, with these 3 base pair substitutions. So we're very lucky that these CArG boxes are very close in space. So with one oligo, we were able to make these 6 substitutions in 2 different CArG boxes. And what we found was that there was a modest decrease at the RNA level, but at the protein level, not much to write home about. And this suggested to us that there must be some redundancy for LMOD1 expression. And in work I don't have time to show you, we've been looking at these other CArG boxes, particularly the one at the 3' end, which has very high binding activity, both in HUVECs and HCASMs, to ask the question whether there's coordinate activity between these intronic elements and the promoter elements that by themselves are not necessary for transcription of LMOD1, at least not substantively. And this is important because at the same time that these experiments were going on, we were knocking LMOD1 out using CRISPR by disrupting the first exon. And what we found was that the phenotype was lethal. It was perinatal lethal. The animals died. They didn't live beyond 5 or 6 days of birth. And a team in the Netherlands, led by Danny Hallam, contacted us and said, hey, we have a family cohort here, an inbred family, as you can see, a consanguineous couple who had some children with something very similar to what we were seeing in the mouse. And long story short, this was published a few years ago, we've found a new disease gene for this thankfully rare disorder called megacystis microcolon intestinal hypoperistalsis syndrome. It's a mouthful, but very, very nasty disease. So it stands to reason that 1 or even 2 transcription factor binding sites would not be necessary for transcription in and of themselves, that there had to be some redundancy lest this disease might be manifest. SRF itself has CArG boxes, and again, the red CArG boxes are conserved, and oftentimes they're conserved all the way down to chicken. And what I want to draw your focus to now is the proximal CArG boxes. So the SRF locus now is on the positive strand, so going from left to right. And in that first, just a 5' of that first exon are several CArG boxes, 2 of which have been studied quite a bit in vitro. But again, we wanted to know whether they were necessary in vivo. And so Qing Liu in the lab designed this experiment very similar to the other one, in that these two CArG boxes are very close to one another. And so one repair template could make these substitutions. So we're very lucky to get several founders, bred them through the germline. And the genotyping of these mice, by the way, it's worth noting, can be tricky. And so Rex devised this method of a duplex PCR assay where he used a allele-specific primer to wild type that had an extended 35 nucleotide extender sequence, and then an allele-specific primer for the mutant allele that was of normal length. And on your right, you can see that we can very easily distinguish genotypes using this duplex PCR system. So very similar to LMOD1, and perhaps not surprising given how important SRF is, there were very modest changes in expression of SRF with these two CArG boxes that previously were shown to be sufficient in a out-of-context transgenic reporter mouse assay system. So moving along the locus of SRF, still in SRF now, there's another, and we defined this very recently in a collaboration with Tom Quatermass at Stanford. There's a very highly conserved CArG box. You see all the way down to a possum, this CArG box is conserved. So we wanted to mutagenize it, and very modest change in the heart. It was variable in the other organs, but not a lot going on. But again, this speaks to redundancy of a very critical transcription factor, absolutely essential for life, and very obviously must be a complex transcriptional regulation involving maybe many distinct transcription factor binding sites, even different from the CArG box itself. And so we're trying to untangle this as well. Okay, so now here's CNN1, calponin-1. This was a gene that I cloned in Eric's lab under his guidance and encouragement. A lot of bad memories with it. But there's a lot that I'm not going to tell you, but just for the sake of time here, we were interested to know whether this specific CArG box in blue was necessary for calponin's expression in the mouse. Okay, so this is a very common theme of my talk. So Yuhan in the lab was charged with making this particular animal model, and she made 5 substitutions in this case of the CArG box. So changing 3 of the initial bases of the CArG box, and created a novel restriction site. For those of you who remember your restriction sites, that's a SAL1 site, very novel. So it makes genotyping very easy, but we opted in this case to use the duplex PCR assay as well. I'm sorry, I had a newer version of the talk, but what am I going to do? I'm not going to go back and start over. So yeah, so we can use a number of methods for genotyping these animals. We can use an RFLP assay, which you'll see in a little bit, or we can use this duplex assay shown here. So this was published. This was actually the first mouse we ever made. We started this work in 2013 when the rocket ship was taking off, and we were using RNA, and it was very difficult just to even get mice at that time. But we managed to get it, and we found this was very stunning, very different than the preceding examples, where here you're seeing essentially a knockout at the RNA level, and even at the protein level. This is Orazio Slavano's work in the lab, and he did this immunohistochemistry, very nice. You see the red, or however color you're seeing it over there. On the top would be wild type, on the bottom would be homozygous CArG mutant mice. So it's almost essentially a knockout by just taking out 5 bases in a regulatory element. Pretty surprising. So we are always asked the question, well, how do you know you're not knocking out other CArG genes? One of the ways we address it, we can talk about this later, time permitting, is to do bulk RNA-seq. And so you can see here calponin is virtually off in the mutants, very robustly expressed in the wild type, but not much else going on. Those outliers are not reproducible. We do our bulk RNA-seqs in triplicate or quadruplicate, and we even repeat the experiment in triplicate or quadruplicate on separate animal tissues. And I can tell you that these outliers in this scatter plot are not reproducibly changed. So that's just noise. The only thing that's changing in these mice is calponin-1. Okay, and so finally, here's another example, and this is from my wife, Xiaochun Long, and she started this work before we were married. Probably we got married because of it. And so she discovered this gene called Tspan2, tetraspanin-2. Incidentally, I'm not giving you the phenotypes because there's really not much to tell about calponin-1 knockout or this tetraspanin-2. So if you have questions about their function, the answer, I'm preemptively answering your questions. The answer is we don't have any phenotypes as of yet, but we're stressing the animals in various ways. So same deal here. So in this case, it's on the CRISPR strand, so right to left transcription. And this is an upstream promoter CArG box, highly conserved once again, all the way down to a possum. Question, is it active? Is it necessary in vivo? And this is just showing that, again, like calponin, that this is a smooth muscle and lyomodin, a smooth muscle restricted gene. And in luciferase assays, you can see very nice CArG dependency in this artificial reporter assay. But what about in vivo? So here's the same deal. This is Peng Gao in Xiaochun's lab who did this experiment. And here is a 3-base substitution in the same nucleotides that we've been targeting previously. And there's the genotyping. And here, like calponin-1, we see a tissue-restricted knockout. So in heart, not much going on. And in brain, not shown here, but shown later, it's most abundantly expressed in brain. And this mutation has no effect whatsoever on those two tissues. And so, in effect, this is, you might consider this a tissue-restricted knockout. And in fact, Xiaochun has proposed that this be considered as an alternative to the more complex construction of Cre-Lox mice, that is, targeting regulatory elements that drive tissue-restricted gene expression to make a knockout. So here are the take-home messages, very mindful of the time here, for part 1. The CArG box, if you studied it carefully, you'll recognize that it's an ideal site for editing because it begins and ends with the classic protospacer adjacent motif, or PAM sequence. And so we've had virtually 100% success in targeting CArG elements with the CRISPR platform. Regulatory genes like LMODI and SRF, absolutely essential, so redundancy. Structural genes like calponin and Tspan2, not redundantly transcribed because they're probably, I would guess, dispensable for human life, since that's the case in mice. Okay, so I'm going to stop here. I'm so used to reading the audience and finding out who's on their cell phones or who's paying attention or who might have a question. If anybody has a question about what I've talked about thus far, we can take a couple minutes and take questions. Otherwise, enjoy the view of the Adirondack Mountains.
Hey, Joe, I got a question.
Yeah, go ahead, yo.
A teleological question. So, as you know, almost all transcription factors belong to multi-member families. What is it about SRF that makes it want to be singular? Is it so resistant to any subtle modifications that you'd see in diversification of the family, or why is it the only member of the family?
That's a great question. I've often wondered it myself. As you know, the SRF belongs to the MADS box family of transcription factors, of which there are 5. One is SRF. The other 4 are members of the MEF2 family, which Eric's lab and others were cloned out and characterized in the early mid-90s, and some labs continue to study those MEF factors. And it's very interesting because the MEF2 binding site bears some resemblance to the CArG box, but it does not bind. MEF2 does not bind to the CArG box, and SRF does not bind to the MEF2 binding site. The answer to the question is I have no idea, and I don't know if it's an experiment that's easily done, because if there are mutations, spontaneous mutations in SRF and loss of function, the animal doesn't even gastrulate. So we'd never know, for example, in the human population, if there were mutations in SRF, we'd never know it because they would miscarry very early in pregnancy, probably, definitely before even a heartbeat, obviously. So, so I don't know. It's a really good question, Eric. The sequence around SRF, those CArG boxes are conserved, certainly in the mammalian clade. You saw with that intronic CArG, it's conserved in a possum. But when you get lower than that, it tends to diverge. So, and by the way, there's SRF in most of the kingdoms of the world, not bacteria or prokaryotes, but in simple organisms, C. elegans, for example, or invertebrates like flies, there's an SRF ortholog, and mutations of it in those organisms is also lethal. So I don't know what it is. It must be that that there is maybe some really good repair processes around that locus to keep it insulated from mutations. But we'd never know that, Eric, right? Because it's essentially impossible to study that. I don't know if I answered your question, but
yeah, you did. I got a comment and another question.
okay.
So, as you know, the MADS box is the primordial transcription factor binding motif that exists all the way back to yeast.
Right.
Although yeast MADS box primordial one is MEF2, not SRF. So I guess SRF evolved when concomitant with the appearance of vertebrate species. Does SRF exist in jellyfish?
I would guess so, but I don't know. If it's in yeast, I would imagine it is. But you're right, that is MEF2. So when did SRF first emerge concurrent with MEF2? That's a good question. I don't know the answer to that. I should know the answer to that.
Yeah, you
I'm
should.
Write it down.
I can't believe you didn't answer that.
You know the answer to it?
I got one last question for you. Are you ready?
Go ahead. Yeah.
As you know, Mike Levine has put forth the concept of shadow enhancers that become active when one enhancer is inactivated. And I'm curious, when you do CArG box mutations in the genome that don't have a phenotype, have you discovered any shadow enhancers that substitute for them?
No, haven't looked. Good question. We're thinking a lot about shadow enhancers with respect to SRF and myocartin, which, as you know, the in vivo regulation of it in the native context is not known as of today. So it's something we thought about but have done nothing to address it.
Thank you.
Okay. So any other questions? That was a great question.
Tony, go ahead.
Feruli there?
Feruli
yeah,
is there.
oh, here I go.
Yeah.
Yeah. Hey, how's it going, Joe? Hi, Eric. So to kind of follow up on what Eric said, I think I am recalling that SRF comes up when mesoderm comes up. You know, that it is, you know, the gastrulation defect may very well be that it's playing a fundamental role in specifying or helping coordinate cells become mesoderm. And like in the single cell organisms like yeast, that's not obviously happening. And so MEF2 is all that is needed.
Thank you for answering Eric's question. Absolutely right. As you know, I'm not a developmental biologist, but that makes a lot of sense, Tony. Thanks for bailing me out.
There's two more questions.
More questions?
Shelley, go ahead.
Thank you, and thank you for the talk. This is kind of a little bit molecular question, and I kind of had two. I was curious if your lab has looked at CPF1, since it is known to cut in a staggered cut versus the blunt cutting that Cas9 uses, if you've looked at that. And then second question was whether or not you've looked at any regions that would be really far upstream. And the reason why I ask this is because working with promoters in my past life, we found one that we were able to make a super promoter by actually increasing the coding length by using what is known as junk, a lot of non-coding sequence that was upstream, that if we included in the creation of this promoter, it actually emphasized its ability and we got increased expression. So I was just curious on those two questions.
So easy first question. We haven't had the need to use CPF1 as of this date. We almost had to use it for a coding mutation I'm not going to talk all about. We've made over 40 strains of mice over the last 8 years, but we've managed to do everything up to this date with just your Cas9, either wild type Cas9 or, as you'll hear in a little bit, the new version of genome editing known as prime editing. with regard to your second question, so we have not. The answer is we have not. There are CArG elements. There are CArG elements everywhere. And one of the things that we're very interested in knowing that speaks to your question, Shelley, is whether distal CArG elements can coordinate a complex that can perhaps span tens of kb with more proximal CArG elements. So we know that most, if not all, functional CArG elements are proximal to the TSS, transcription start site. To my knowledge, no one has demonstrated a functional CArG element that's more than 4 or 5 kb from the TSS. That's not to say there isn't one. So we haven't done your experiment, but we're mindful of the fact that there are highly conserved CArG elements that are in some cases hundreds of thousands of nucleotides away from its nearest annotated transcript. Is that functional? And this is a question, sort of a pet question of ours that we're curious about. So no, we haven't. We don't want to. We want very clean experiments when we're doing these editing experiments. So they're all done in their native context with no molecular gymnastics, if you will, that we all used to do back in the day. I think those experiments at that time did inform us of things, but right now we're in the real world with this very powerful technology, and so we're making edits where the element lives under normal circumstances and without any other adulterations to the locus. So perhaps I should move along.
Yeah, makes sense. Thank you so much.
Yeah, sure. Maybe I should move along, and if there's time, I'm happy to stay on as long as everyone's willing to listen to me talk about this. But now I want to change the subject a little bit, not a lot, to SNVs or CArG SNPs. So I've been interested in this for the last 15 years or more, but I don't know bioinformatics. And so Craig Benson is still a bioinformatician. He went to the dark side. He's in industry now. But he did this experiment published several years ago where he first defined CArG variants that exist in people, and then he modeled these in this gel shift assay. And you can look, not a lot of explaining here, kelponin is our control, and you can see in most cases, SRF binding was perturbed, in some cases virtually abolished, by a single nucleotide base change, right? And so the question, of course, is, does that have any meaning in our life, in biology or pathobiology? So in more recent years, and these are data that are probably about 2 or 3 years old now, although they've been updated, because I see the DB SNP is build 153. Di Huang and Ivan Avchenko's lab at the NCBI surveyed the mouse and human genome for all CArG variants, and astoundingly, the number is nearly 1 million. So there's 1 million theoretical, probably actual, CArG boxes that have one or more variants in them. Most of them are rare, and many of them are common. And this is the distribution in the graph there across the 10 base pair element of rare and common variants. And so this is an astoundingly large number, right? So we're not going to make 1 million CArG box mutant mice ever. So we have to try to figure out how to do this smart. So Mike Wilson in the lab devised this experiment, a cold competitor experiment, where the question was, which nucleotide in the CArG box is least tolerant to change, right? So this, so the weaker the signal, the more tolerant the change, right? So in the first blue circled variant, that first position, that transversion C to G, there's not much competition with cold competitor, and so this substitution is not tolerant to change. And so we use this information to inform how to proceed with trying to prioritize things with respect to CArG SNPs or SNVs. So we wanted to do some proof of principle experiments, and so Qing Liu in the lab went back to the kelponin locus and I talked about this earlier with a 5 base pair substitution, and he remade the mouse using the same protospacer and the same oligo, only now he made one base transversion, a C to G transversion, and that fortuitously for us created a SAC2 site, which really simplified genotyping as shown in this PCR-RFLP. And he found essentially what was reported in our ATVB paper several years ago, which is a one nucleotide substitution essentially abolished expression in a tissue-restricted manner. So not much in the bladder, a little bit, but in the aorta and presumably other vessels, very little in the way of expression. And this is shown quite provocatively here in this immunohistochemistry experiment. CNN1 is in red, and then there's actin, smooth muscle alpha actin in green, coincident yellow on top in the wild type, but in the bottom mutant in these different vascular beds, you see essentially no kelponin protein. So this, again, highlights what I mentioned earlier, that this is a way, we've found a way, at least in some cases, to make a tissue-restricted knockout by changing a single base in the genome, which is really unprecedented. I've combed through the literature and talked to the experts, and no one has been able to present me with a precedent for such a finding. I'm happy to be wrong, by the way. The paper is still under review, so I can change it and reference. If anybody knows of an exception, please tell me. So we thought at that time that this was a one-off and it couldn't possibly be true of other CArG elements in other gene loci. And so here's a gene that we studied several years ago and reported on smooth muscle gamma-actin, again, tissue-restricted for smooth muscle. There's several CArG boxes in the 5' promoter region, one of which captured our attention based on the luciferase data in the lower right here, where if we mutated one of those CArG boxes, we essentially abolished luciferase activity. So that informed us as to which CArG box among these 5 to select for further study. And so Mary Wine-Samuelson in the lab made this one nucleotide transversion in the gamma-actin locus. And again, same thing. So here in vascular aorta, smooth muscle cells, essentially a knockout. In other tissues, either not much going on at all or very little. The uterus result, I get asked that question a lot. In repeated experiments, it's not knocked down that much. Okay, enter prime editing, all right? And this brings us around third base. So this was published by David Liu's lab a year ago, yesterday, Wednesday? And so David Liu, for those who don't know, is one of my heroes, CRISPR heroes. He developed a number of platforms, including base editing, which I was very interested in using for CArG box editing, but because of the CG-rich nature of CArG boxes, it wasn't so precise. When this paper was published, we got really excited, and we jumped on this immediately for making CArG mutations. And so what I'm going to share with you today is unpublished data that's under review now at Nature Cell Biology, and it basically is a comparison of traditional 3-component CRISPR with prime editing. All right, so who wins? So for those who don't know about this, first, there's the paper. I invite you to read it. It's a bit complicated, but David's brilliant. So the story goes, he was walking in Cambridge and was pondering, how do we make the guide RNA more versatile? And so he came up with this idea of fusing Cas9 to a reverse transcriptase and then extending the tracer RNA discovered by, or I should say, formed by Jennifer and Emmanuel in 2012, extending it with this template. So a primer binding site and a reverse transcriptase template that would include the edit, your favorite edit. In our case, it would be a single base substitution. And the idea is that this Cas9 RT fusion is a nickase, so it only nicks one strand of the DNA and allows the primer binding site to engage the nick, and the reverse transcriptase then uses that as a template to write in the edit, or in this case, the single base pair. But he showed insertions, deletions, and so on. I think he did like dozens and dozens of different substitutions. So every conceivable substitution is possible with this, not true with base editing. And so this is a very powerful platform with a lot of hope for the future. And so this is just to remind you in a little more decorative schematic of what Xiaoshun showed a few years ago, which is that a 3-base pair element, a 3-base pair substitution in this CArG box creates essentially a knockout. It's shown here. These are new experiments now These are the data that have been submitted. So you see an aorta bladder, essentially gone. But in brain, where it's highest expressed, nothing going on. In a heart, which is intermediate in expression, nothing going on. So this is just to remind you of that edit. And so we wanted to compare the this three-component CRISPR editing platform directly, head-to-head, with the prime editing platform of David Liu's. So the first thing we did after David gave us the plasmid was we had to figure out how to get conditions down for transcription of this 6 kb plus transcript, right? So Amr Ghanem in the lab, very passionate guy and talented, came up with the conditions that gave us very nice transcripts you see on the right here so that we could inject, because unlike Cas9, there is no Cas9 NRT protein as of yet. So this is an RNA, an mRNA that we have to co-inject with a PEG RNA and the Cas9 NRT editor. So I work a lot with Synthago. My good buddy Kevin Holden is senior scientist there, and Synthago, the first PEG RNA they synthesized was ours back in early December of last year. And the PEG RNA is as follows. So it's the same protospacer as the sgRNA that I showed you earlier for Tspan2, targeting the CArG box, and of course the tracer RNA. But here, and we guessed, I have to be honest, I get asked the question often, did you test multiple PEGs? The answer is no. We looked at all the data that David provided in his supplement, which numbers, I don't know how many dozens of pages. I don't know if people have pored over it, but we have. And we just best guessed, based on all the data, we chose a primer binding site of 16 nucleotides. Those 3 U's are added at the end from Synthago to stabilize it. And then the RT template was about 10 nucleotides with the single transversion, the G residue in it. And so the question was, will this work? Will we get precision editing? I was worried that the RT would just continue right along into the scaffold and we'd get all this scaffold and stuff. So here's the genotyping. So that transversion creates a novel restriction site that we exploited for genotyping purposes. And there's the sequence. We were able to install this transversion pretty well from the Singer sequencing. We had pretty nice turnout with a number of founders that were positive. And here's the result. Perhaps not surprising based on the kelponin example I showed earlier. This single base edit in the mouse with prime editing essentially created a knockout, a tissue-restricted knockout of Tspan2 in the mouse. Now, there's an overlapping, in the mouse, there's an overlapping link RNA, which is another big part of my lab, and I'll only mention link RNAs in this breath, and Tspan2 OS for opposite or opposing strand. And remarkably, but maybe not surprisingly, this single base edit controls not only Tspan2, but tetraspanin2 as well. So essentially, tetraspanin2 OS is knocked out. So at that point, well, not at that point. I mean, the history was we called David immediately when his paper came out. And so this is a collaboration with the Liu lab. And so Greg Newby in David's lab was very kind to do this on-target editing efficiency, both with the 3-component CRISPR, which I'm showing now, and then the prime editing I'll show in just a moment. So there's a lot of data here, but what you'll see in the graph is very nice, desirable editing, very high, which is typical for CRISPR-Cas, but it comes at a cost. So the gray colored bars represent the percent alleles with indels. And so we had a percentage overall of about 50% correct editing in a range of 1.67 to 95 plus. But the cost was there was a fair amount of on-target indels, which if one is thinking about somatic editing, which is really our end game that I won't talk about today, and utero genome editing is one of the things we're working on in the lab, is not a desirable thing to have happen. So in contrast, in sharp contrast, with this locus, under these conditions, there was not so great on-target correct editing, desirable editing, but reasonable. But more importantly, no detectable on-target indels, none, 0. So very, very precise editing, only that single base, and we were really delighted. But what about off-targets, right? So this was and by the way, this is all work done in collaboration with other labs. So this is Shengdar Tsai at St. Jude's and Cicero Lazzarotto, who published recently this change-seq, which I don't know if anybody's looked into it, but we're working with the Tsai lab now because this is really a powerful way to look at off-targeting. So Cicero looked at the guide RNA and the PEG RNA, compared them head-to-head. Now, necessarily here, and I don't have time to go into what change-seq is all about. I urge you to look at the Nature Biotechnology paper from Cicero and Tsai's lab for details, but there was no way to use the Cas9nRT protein in the change-seq because it doesn't exist as a protein. So she had to use Cas9. So she compared the PEG RNA and sgRNA with Cas9, and for reasons we don't understand, the sgRNA, probably structural, the sgRNA gave more predicted off-targets with the change-seq than the PEG RNA. This is the important experiment on the bottom. I'm skipping a lot of data, but these are Manhattan plots showing the extent of off-targeting genome-wide with the guide RNA versus the PEG RNA. And you can see, although it's not dramatic, it does make the point that there's more off-targeting, much more off-targeting with the sgRNA than there is with the PEG RNA. And by the way, these data below reflect, again, I'm skipping a lot of data in the interest of time, they reflect an analysis of 11 founder mice in the sgRNA arm and 12 founder mice in the PEG RNA arm. So this is collapsed data, a lot of data, reams of data. And I can tell you that 5 of the 11 sgRNA founders had off-target indels. 0 of the PEG RNA founders had any off-targeting events. So it turns out this is a very, at least for this locus, a very useful platform for making precision edits. So I'm wrapping things up now. Here are the take-home messages for part 2. And this is an astounding statement. And we have more examples coming, by the way, so stay tuned for those. But a single base substitution of a transcription factor binding site abolishes target gene expression. This has profound implications for not only the CArG Variome, as we like to refer to it, but other transcription factor binding sites where single base substitutions may have profound effects on target gene expression. The prime editing version 2 platform, there are different platforms, I didn't have time to go into that, yields perfect editing at the Tspan2 CAG box with minimal, essentially nothing above background off-targeting events. And the PE2 will be helpful in correcting and modeling. And we're using the PE2 model system now to not only model mutations, but correct them as well. And this is all being done in animal models with real CArG variants in people. But I will reserve those comments for another date. So I want to thank a lot of people from my lab, my wife's lab, Xiaoxun Long, Shengdar Tsai, who's been really helpful, David Liu, who is just an amazing character in the field. John Chimenti was huge because as we did all this while we were moving and during COVID, which really is a remarkable feat and really demonstrates the value of collaborating with multiple labs, because obviously we could never do this alone. And then, of course, Synthago, Kevin et al. For their PEG RNA, and very generous funding from my current institution and the NIH and American Heart and Synthago. And here is the last slide before I open up things for questions. I always show this slide. I don't know, I've given like 40 plus CRISPR talks over the years, and I always show this slide because I hail all these people. I mean, Jennifer and Emmanuel will be the first to say that this has been a global effort. Lots of talented people, including people who did editing decades ago. The Maria Jazzens of the world, the zinc finger nuclease people of the world, Dana Carroll and Matthew Porteous, very a good friend of mine, Fyodor Urnov, who's one of the most articulate scientists I know on the planet Earth. And by the way, he's in the movie Human Nature. Keith Jones, so many people to thank for developing these platforms that have made my life phenomenally easier and very interesting. And so with that, I'll stop and try to answer any questions you might have.
And I'll monitor the room. If you could raise your hand, are there any questions for our speaker?
And Eric and Tony can ask me more questions if they want.
Eric, go ahead.
I have a question. Great stuff, Joe. So did you detect any functional consequences of the off-target edits with prime editing, and where were they localized in the genome?
So with prime editing, we didn't see anything above background in the way of off-targeting. With respect to sgRNA off-targeting, nothing to really write home about, Eric. And because essentially all of the off-targets, actually all the off-targets in the sgRNA arm were on other chromosomes, they were likely bred away. We're now in like the third, fourth generation of these animals, and they're perfectly fine. They breed, as do the kelponin single base edits breed, as do the ACTG2 animals. They breed, they look fine. But where we're at now, and this is very early days because we're making so many of these CArG mutant mice, is to stress the animals in a variety of ways. So like blood pressure, challenging the animals with a variety of agonists that would elevate their blood pressure or create a lesion in their vessels and so forth. Chronic conditions like athero. So we're at that stage now with Tspan2, for example. So the answer is none with prime editing. We haven't done any other loci with prime editing, though we're gearing up to do it. That will be our go-to now for making these single base edits in CArG boxes that exist in people, by the way. So I should mention, I often get asked the question, are these mutations, these substitutions, do they exist in people? The answer is no, they don't. In the case of Tspan2, CNN1, and ACTG2, those are contrived. They're just proof of principle experiments. The real CArG SNVs are breeding now, and we're just waiting to see what the phenotypes might be. So that's all I have to say about that.
Thank you. Great stuff.
Any other questions?
Must have
So
been pretty clear.
I'd like to make a comment. Your single nucleotide mutation that knocks out expression of a given gene in a specific tissue, it's the first time I've ever seen anything like that. It's stunning. So what it suggests to me is that SRF and a given CArG box function in a tissue-specific manner in concert as an integrator of the milieu of whatever co-regulatory proteins are expressed in bladder or smooth muscle per se. So that's just a phenomenal result. Thank you for sharing that.
No, you have a question in there though, because I get asked that question often. How does that work, right?
Yeah.
And you really can't invoke myocardium. I mean, you can to an extent, but in the bladder, there's lots of myocardium in the bladder. And so you would expect myocardium to collaborate with SRF over those CArG boxes to drive expression. And if you muck up that binding site, then you're going to muck it up everywhere there's smooth muscle. And that's certainly the case with Tspan2, but not true with gamma-actin or kelponin 1. So there's more to the story than SRF myocardium. Now, is there a composite element? Is there something neighboring by? It could be distal. To Shelley's question earlier, there could be something distal that's looping back onto it in the bladder, for example, that's unique to bladder. We don't know. I mean, those are questions that are outstanding questions. We're not pursuing them, but they're very good questions. So your comment, it was actually a comment question.
Let's go fishing with yeast hybrid. By the way, I still have that rat aortic smooth muscle cell library that you made in my freezer. Just so you know.
Yeah. And myocardium wasn't in there, Eric. We had we didn't have the computer power that Daji had. We just had the wrong libraries to. If we had a primary cell line, perhaps we would have fished out myocardium. But we had those stable cell lines or multiply passage cell lines, and myocardium goes away pretty quick in culture. So I'm not too sure those will be useful for you, Jeff. Those
That's
cells.
A great library, at least on a gel. Tony, go ahead. You got a question?
to follow up on what you guys are talking about. So it's pretty clear, probably, if not definitely, that like SRF is a pioneer factor, right? So when you talk about this single base substitution, having such a dramatic effect, have you ever just done ATAC on the mice that have and have not the mutation to just see whether or not, DNA access is altered, or think about just looking at 4C or something to see whether you're changing a TAD? Because it seems to me that you're doing something more significant than just inhibiting a factor from binding DNA, and that the consequence of that DNA binding is topology driven.
Yeah, no, Tony, that's a really great question. I've never been asked that question. It's outstanding. We haven't done ATAC-seq. What we're gearing up to do in human cells, because the whole CArGome odyssey, it really truly is an odyssey, is steered towards the human condition, right? So all these variants, and we've got hundreds of them that we're trying to prioritize in a meaningful way. We really want to stick with human, and we've done ChIP-seq. We're doing H3K27 acetylation. I like the idea of ATAC-seq though. But your question, the closest thing I can think of that would address your question, Tony, and it's not going to be satisfactory, is the acetylated status over, I believe, Tspan2. One of the three, I can't remember which one, is not changed. So in other words, it is, at least for that chromatin mark. But the ATAC-seq or some other means of looking at the openness is a really good idea. And we really should do that. And I wrote it down with stars around it, Tony. So thank you for asking that.